Hello everyone. In the previous class we discussed about structure of Golgi bodies. In this session we will discuss about functions of Golgi complex or Golgi bodies. The main functions of Golgi bodies will be discussed in this class. In the first one is secretion and packing or packaging. What are secretions? Some substances released by cells or glands, that is called secretion. Secretions are produced by proteins. They are produced by proteins. Where the proteins are present? Proteins are present on the endoplasmic reticulum, rough endoplasmic reticulum. On the endoplasmic reticulum, proteins are present. They release some substances. After that, for example, this is rough endoplasmic reticulum. Okay. From this rough endoplasmic reticulum, some vesicles are produced. On the surface of it, ribosomes are present. Those ribosomes releases secretions are concentrated in the endoplasmic reticulum. Further, vesicles are budding up from the endoplasmic reticulum. Then vesicles are separated. These vesicles are now coming to contact with cisternae. These are cisternae. These vesicles come into contact with cisternae, and the products of these vesicles are entered into the cisternae. In the cisternae, they are concentrated. After some period of time, what happens? The ends of the cisternae swollen in their size and they are pinched off. Then they are called vesicles and they contain these secretions. They may be in the form of hydrolytic enzymes, hormones, or other secretions. Okay. Some examples for secretions are, for example, hormones. They are useful in the secretion of hormones. Example, thyroxine. Thyroxine is a hormone. In the secretion of thyroxine, Golgi bodies are useful. Next one is collagen. Collagen is secreted by connective tissues. And one more, polysaccharides. These polysaccharides are secreted by goblet cells. Okay, are you understand? And exocrine glands. These exocrine glands releases or secretes enzymes. These all are secretions secreted by Golgi bodies, and they are packed in the form of vesicles or etc. Okay. Next, one more example, proteins also secreted by some ribosomes or endoplasmic reticulum or Golgi bodies. Examples for protein secreting cells are achinal cells of pancreas. This is about some secretions. Next, coming to another function, that is formation of acrosome. That is also one of the important function of Golgi bodies. Acrosomes are present at the tip or top of the sperms. Okay. For the formation of acrosome, Golgi bodies are very useful. Next function is third function. Golgi bodies are useful in the formation of cell wall. Formation of cell wall. Cell wall. During cytokinesis, what happened before the cytokinesis? Karyokinesis takes place. For example, this is mother cell. In the mother cell, after karyokinesis, two daughter nuclei are formed. In between two daughter nuclei, cell plate is formed. Cell plate is formed. Golgi bodies are accumulated on equatorial plane. They are accumulated in the equatorial plane and they forms cell wall. They forms cell wall. And they are also useful in the formation of middle lamella. Formation of middle lamellum. Middle lamellum. Generally, this middle lamellum is made up of with calcium and magnesium pectates. Calcium and magnesium pectates. These calcium and magnesium pectates are secreted by Golgi bodies. Or understand? They are involved in the formation of cell wall in the plant cells, and they are also involved in the formation of middle lamella in the plant cells. In middle lamellum is generally made up of with calcium and magnesium pectates. They are secreted by Golgi complex or Golgi bodies. Next function is Golgi bodies are useful in vitellogenesis. Vitellogenesis. Vitellogenesis is nothing but yolk formation. Yolk formation. In yolk formation also, these Golgi bodies are very useful. Next, another function of Golgi bodies is they are involved in GERL system. GERL system. GERL system. G, it is nothing but G means Golgi bodies, ER means endoplasmic reticulum, L means lysosomes. How the substances are moving from Golgi bodies to endoplasmic reticulum and endoplasmic to reticulum to lysosomes, that will be explained in the later classes. In that transport also these Golgi bodies are involving. Okay. Next function is glycosylation. Glycosylation. Glycosylation is nothing but addition of carbohydrates to proteins or lipids. Addition of carbohydrates to proteins or lipids. Proteins or lipids. If the carbohydrates are added with proteins, carbohydrates plus proteins, then they form glycoproteins. They form glycoproteins. These glycoproteins are useful in identification of cell. Identification of cells. Cell identification. Okay. If the carbohydrates are added with lipids, carbohydrates are combined with lipids, they form glycolipids. Carbohydrates plus lipids, they form glycolipids. Glycolipids. These glycolipids are very useful in protection. They are useful in protection. I understand? Protection. Next, one more function is there for Golgi bodies, that is eighth function. The eighth function is formation of primary lysosomes. Formation of primary lysosomes. Primary lysosomes. Lysosomes. In the Golgi bodies, cisternae are present. These are, for example, cisternae. The ends of the cisternae are called Golgian bodies. When the substances or material is stored in the cisternae, after some period of time, exocytosis takes place, resulting in the formation of a bud-like structure originates from the cisternae, and they are pinched off from cisternae or budding off from cisternae. Then these structures are called primary lysosomes. These primary lysosomes becomes lysosomes after some period of time. They are called lysosomes. They have hydrolytic enzymes. In their formation also Golgi bodies are involving. Okay, this is about functions of Golgi bodies. In the next class I will come with another cell organelle. Thank you.